right, folks, I'm ready. How are you guys today? Fun stuff. I've talked with you to death about chemistry, talked with you a fair bit about cell communication. That was last Thursday. Now we get to cell division, and that sets the stage for me to talk with you Thursday and Tuesday about some genetics stuff. We'll finally start getting into inheritance and how information gets passed from one generation to another. Again, we're going to set the stage for that today. Today I want to talk with you about cell division. That's the main topic. But before we really get to that, let me remind you about what's hanging behind me on the screen here this morning. And there's going to be another test in this class. All right, that's not a big surprise. It's going to be on Thursday of next week. It's going to be right here in this room for those of you who are here right now, I presume. If you're one of the students who are just listening to these recordings on Integrity, then there's a separate room that we have for you. That's 101 Fawcett. As with the first test, if you have a conflict, particularly for the students who are doing the online version of the course, if you've got a conflict that prevents you from being able to be here for the test itself at those times during the class meeting time, let one of the supplemental instructors know, particularly Lauren Trout, but Haley will be able to help you as well, and we'll be able to let you take the test 1 hour earlier to fit better with your schedule. So just like with the first test, if you are needing to take the exam at a different time, contact the supplemental instructors for the course, and we'll figure out a way for you to take it earlier than the rest of the class. But for the rest of you who don't have to worry about that, instead of class next Thursday, all that's going to happen is you're going to get a second test. I really cannot emphasize it enough. I'm sure you're sick of me saying this. Look at the old tests. Okay, that's going to be your single best way to get ready. This is plenty soon enough for you to start looking at those old tests. Don't worry that you're going to peak too soon or something like that. Start looking at them now, and that'll help you key in on what it is that you should be focusing on for the second test for this class. All right, so look at the old exams. At the start of the semester, I think we had 4 old exams, old exam 2s, on the pilot page for the course. Since then, another 5 or 6 have been added. Here's the thing. The first 4 that are up there are from when this course was offered as part of a semester-long class as opposed to a 1-quarter class. Wright State made a transition from quarters to semesters about 4 or 5 years ago. I hesitate to put those older exams on the pilot page because there were some differences in terms of the way the course got broken down and the things we talked about when this course was on quarters as opposed to semesters. So if you're looking at these old tests for a guide as to the kind of questions, what sorts of things I'll be asking questions about, it's those first 4 that are going to be your best guide. If you're just looking for lots of questions, some of which may not be part of what's been covered for this version of the course, then there's some older ones that you can look at as well. The older tests are like 80% the same material. The newer tests are 100% the same material. So just keep that in mind. On those older tests, for instance, there's nothing about cell signaling. Cell signaling only got talked about in this class when we switched to semesters. All right? So don't freak out, and bear in mind that's why those 2 tests may talk about different sets of things. My rule is that if I don't talk about it during the course of a lecture, I don't want to ask you about it in an exam. So if you see some questions and you say, gee, I never heard that get discussed in class, you don't have to worry about that question for the test. That probably just means that particular year, in particular if you're going back 5, 6 years, that just means that I was talking about some things that are no longer talked about, at least at this part of this class. Okay? So I really can't encourage you strongly enough, look at those old exams. I think that's going to be a big help to you. The test is going to be 45 multiple choice questions, just like all the ones you see on the pilot page for the course. Bring a number 2 pencil. You'll need it. You're going to fill out those Scantron forms. And our goal will be to have grades posted on Pilot by the end of the day on November 3rd. We'll see what we can do to pull that off for you. All right? So enough about that stuff. Today, again, I want to start talking with you about cellular reproduction. And I know in lab a week or 2 ago, you did all this stuff, and so this is going to be easy for both you and for me, right? This is just going to be a review of some of the things you did in lab, maybe giving you a little bit of my spin on things that you were seeing when you did this with a microscope in lab. And let's start out by just having me remind you kind of what the point of all this is. And I keep coming back to that first lecture. When we first started talking about this class, I said, biology is the study of life. What's it mean to be alive? How do you discern? How do you decide if something's alive or not alive? And one of the things on that list of 4 or so criteria that I threw out to you was be more organized than your surroundings, right? That's pretty much all that I've been talking with you about ever since. All this energy stuff, all this metabolism stuff, those are all things that are related to how it is that living things can be more organized than their surroundings. When I started talking with you about cell signaling, we made a little bit of a change. We're no longer talking just about that one criteria. We're talking then also with the cell signaling about responding to stimuli. Okay, that was one of the other 4 criteria that I said you should expect to find with a living thing. Being able to respond to a stimulus, being able to change what you do because of some change in your environment. And I'm going to talk with you about that a lot more after that second test when we talk about how genes get turned on and turned off. Boy, that is easily one of my favorite things to talk about. You're going to have a hard time getting me to stop talking about those things. It's going to be bad, I warn you right now. But today, we're touching perhaps the only time on a third of those 4 things that I said distinguished living things from non-living things. Maybe you could say I'm touching on the 3rd and the 4th. One of those was the ability to repair damage. Cellular reproduction has got to be at the top of the list for repairing damage, and also the ability to reproduce. So that's the 4 things that I said you might expect to find a living thing to be able to do, whereas a non-living thing would struggle. The soapbox derby's at it again. If you're curious as to what's going on out there, we'll set up like a live webcam someday, and we can watch what's going on while I'm lecturing. But so we got this ability to reproduce and this ability to repair damage. You can't repair damage very effectively as a living thing if you can't have cells divide to fill in the gaps and take the place of the damaged parts, right? And so that's a big part of today's lecture. And then also this ability to reproduce. What I'm going to talk with you about today is mitosis, and then later on Thursday, I'm going to talk with you about meiosis. That's going to be the reproductive part of it here, but you can't do meiosis unless you've got mitosis in place. And so we're kind of laying the foundations for that ability to reproduce things too. So anyway, if you're keeping score, if you're trying to keep track why this stuff matters, it matters because this is a pretty big part of being alive, right? And it's a big part of being alive if for no other reason than it's pretty much the only way that we can go about repairing damage. But it also lays a framework that we can use for sexual reproduction with meiosis. And we'll talk about that on Thursday. So let's start at the beginning, right? This idea of prokaryotic cell division. Once again, prokaryotes, if I say prokaryote, I hope one of the first words that pops into your head is simple. If I say eukaryote, I hope one of the first things that pops into your head is complex or complicated. So often, we find that biologists figure stuff out by studying these simple prokaryotic systems, and that gives us important insights in terms of what's going on with the more complicated eukaryotic systems. So, in that same spirit, let's start with the simple prokaryotes, and then we'll work our way up to the more complicated eukaryotes. Prokaryotic cell division is a fairly simple thing, okay? Not a lot of complexity here. Remember that picture that I tried to get you to put in your heads about what a prokaryotic cell was? I would expect you'll remember I used the phrase, prokaryotic cells are essentially a bag of goo, right? What's in the goo? The goo has some proteins, the goo has some sugars and some starches, the goo has some DNA, okay? That's the goo. What's the bag? It's the cell's membrane, okay? What do you suppose you need to do to get one bag of goo into 2 bags of goo? Well, nothing too complicated here, right? You probably need to eat some stuff, right? So if a prokaryote wants to become 2 prokaryotic cells, job number 1 is going out into the world and getting the resources that it needs to make more of its goo. Right? More of its proteins, more of its sugars, more of its membrane, more of its DNA. Once it's gotten enough resources such that it's got twice as much stuff as what it started with, that's the time for this cell to divide. And it's pretty much as simple as that. So let's take a look here at what's going on. You start out with a single cell. Here we've got a picture of an artist's rendition of an E. coli cell. You start out with a single cell. The cell makes a copy of its DNA molecule, gets bigger, and ultimately splits into 2 cells, each with 1 copy of its chromosome inside of it. So that's it in a nutshell. Perhaps the most important part of prokaryotic cell division is this business of replicating the bacteria's chromosome, okay? A little bit of jargon for you here. You started out with 1 cell, you end up with 2 cells. Those 2 cells are called daughter cells. They're sisters to each other, but they're pretty much indistinguishable from each other, right? They're effectively perfect copies. They're indistinguishable copies of each other. And they can't do very well unless they're complete and a perfect copy of their genetic instructions. Those genetic instructions are stored on a chromosome. For E. coli, it's a single chromosome, 1 big old circular DNA molecule. When I say a big old circular DNA molecule, let me be a little bit more sciencey, right, and tell you that big old circle is 4 million nucleotides long. 4 million G's, A's, T's, and C's strung together in a particular sequence that together spell out all the instructions that E. coli needs in order to make another copy of itself. Its DNA, its membranes, its proteins, the whole shebang. It's the blueprint for that E. coli. 4 million letters, 4 million nucleotides spell out those instructions, and those instructions are preserved and faithfully passed on from one generation to the next on a single circular piece of DNA, okay? How long do you suppose that process takes? I'll tell you the rate-limiting step for E. coli to go from 1 cell to having 2 perfect copy daughter cells. The rate-limiting step is the time that it takes for E. coli to make a copy of its DNA molecule. There are other times that other things can be rate limiting, like how much food's available. But if you take care of all the rest of E. coli's problems, the one thing that it can't do any faster is how long it takes to replicate its DNA molecule. The time that it takes for E. coli to replicate its DNA molecule is about 30 minutes. So you can double the number of E. coli cells in a test tube or on a Petri dish once every 30 minutes, and what you're waiting for is the time that it takes to make a perfect copy of that 4 million nucleotide long DNA molecule. That's a pretty good rate of growth. That's exponential growth. I mentioned that in the lecture on Thursday, the difference between exponential and lag phase growth. Exponential growth, every 30 minutes you double the number of cells that you're dealing with. Not bad if you can pull it off. Pause for a moment and think about the logistics of what's going on here. There are two numbers that I think it's fun to just wrestle with for a little bit. One of them is, again, this 4 million number. That's how many letters are in the E. coli genome. And the other is 30 minutes. What I'm telling you, put those things together, what I'm telling you is that at some level, E. coli needs to make a perfect copy of all 4 million nucleotides in as little as 30 minutes. If you do the math associated with that, that works out to about 2,000 to 2,500 nucleotides copied per second. Whoa, right? Is that impressive or what? That's no mean feat to be able to make a perfect copy, error-free, 2,500 letters copied per second. And yet that's what's happening when E. coli commits to making a copy of its DNA and dividing a cell into two daughter cells. Pretty slick. Hard to even imagine how something could be done that quickly. 2,500 letters per second, right? Not per minute, per second, right? Hard to imagine something could work that fast, let alone be that accurate. Absolutely perfect, no mistakes, no mutations, because that's what a mistake would be called, by the way, a mutation. Amazing stuff. In about 2 weeks, I'm going to take you by the hand, and I'll get you at a molecular level, show you how it is that those copies actually are being made. For now, just be impressed. Maybe look forward to seeing how it is that it actually gets accomplished. But appreciate that that's pretty much what's holding E. coli back. If you didn't have so big a genome, if you could copy that genome more quickly, those are the only ways that E. coli could be faster and better at making copies of itself than it is. So job number one is getting bigger, getting the resources you need that would allow you to make a copy of your chromosome. Job number two is making a copy of that chromosome so that you can divide into daughter cells that, when you look at them, they're indistinguishable from each other. Here is an actual picture of the process playing out. These are real cells that are doing the business of division. You can see this was a rod-like cell that became longer than its neighbors. Maybe to some extent you can see that. And the DNA here has been stained so it pops out and you can see it a little bit more easily. And you can see the cell is dividing. It's constricting here in a way that's going to cause you to get one copy of the cell's chromosome on one side and the other copy on the other side. So, you know, the cartoons that we've been looking at, not that different from what's happening in real life. This is simple, right? Not that much going on here. You got a bag of goo. You make a copy of all the stuff in the bag of goo. You pinch the bag of goo in the middle, and you end up with two smaller bags of goo, right? That's what's going on. Nothing more to it than that. Let me draw your attention to one other thing that's kind of a big deal. It's a much bigger deal for eukaryotes, where we're going in just a minute. What if this doesn't work out right? What if that pinching, and let's be fancy and call it a cleavage furrow. What if the cell's cleavage furrow happens in a way that doesn't get you a copy of the chromosome on both sides of the cleavage furrow? What if there's a mistake? How bad is it if instead of getting two daughter cells that each have a copy of the chromosome, you end up with a daughter cell that has no chromosome and a daughter cell that has two chromosomes, right? How bad is that? You know what happens to the cell without the chromosome, the daughter cell that didn't get a chromosome? It fiddles away, right? It's got no capacity to respond to changes in its environment. It can't make more proteins. When its proteins wear out, it eventually breaks down, falls apart, goes away. It's effectively dead. It may take a day or two for that to happen, but not much. It's a dead end. Nothing's going to happen of consequence with that daughter cell. You know what happens to the other daughter cell, the one that got 2 copies of its chromosome? It just got a 30-minute bonus in terms of how much time it needs to divide again, right? Because it's already got 2 copies of its chromosome. It doesn't need to replicate that DNA anymore. Now all it needs to do is find enough food to let it get enough mass to make more membranes, more proteins, and get ready to do this process a second time. Short answer, how bad is it if you fail to do this correctly? It's not that bad, right? It's certainly not the end of the world. It's not a absolutely catastrophic event for the E. coli. It's not life or death. One of the 2 daughter cells is going to be just fine. The other daughter cell, well, that's just a price you pay for having made the mistake, I guess. But, you know, the species lives on, right? So it's not that bad if this doesn't work the way that it should. The reality is it seems to work the way that it should much, much more often than not. But that's what's up with prokaryotes. Again, fairly simple. Big part of it is just getting bigger, and a big part of it is replicating the chromosome. Beyond that, pretty much everything else is details. Okay? What I've just described to you actually gets a fancy name. So if you want to be fancy when you're talking to biology people about prokaryotic cell division, you could call that process binary fission. Okay? Binary, because you're making 2, fission, because you're splitting it into 2. Right? So this is all binary fission, and not much more to it than that. Well, let's change gears a little bit and talk about what's going on with eukaryotes. So that was the simple part, lays a foundation, the basic principle, pretty much what you would have expected. Nothing fancy going on there. Let's change gears and talk about what's going on with eukaryotes. Right out of the gate, eukaryotes are more complicated in 2 big ways relative to prokaryotes for cell division. Let's do the one that's consistent with what we've been talking about all along so far. Eukaryotes have those pesky internal membrane-bound compartments, right? It's not enough to just get bigger as a eukaryotic cell. You got to get, when you're getting bigger, you also have to make sure that both of the daughter cells get a complete set of those internal membrane-bound compartments. If one of the daughter cells doesn't get a mitochondria, you're screwed. That cell it's got no future. If one of them doesn't get a chloroplast, it's got no future, right? So you have to get these internal membrane-bound compartments equally apportioned. But included in those internal membrane-bound compartments is something that's leading me to the second problem. Included is this idea of a nucleus. Because what's the nucleus again? That's where all the DNA, that's where all the genetic instructions are bundled up and stored. So you got a wrapper, and not just a wrapper, a double layer wrapper around the stuff that you need to replicate and then make sure it gets to both sides. So that wrapper has got to go away at the very least. How are you going to pull this off without unwrapping the DNA in the first place? So that nuclear membrane is going to be a problem. And what's inside of that nuclear membrane is also a problem relative to what we saw with prokaryotes. With E. coli, it has 1 circular chromosome, 4 million nucleotides long. Sounds like a lot. Not, however, when you put it in the context of what's going on inside of your cells, inside of your nuclei. Your genome isn't 4 million nucleotides long. It's bigger. It's a lot more complicated. You know how much bigger? Like 1,000 times bigger. Your genome is 3.2 billion nucleotides long, right? So replicating all that DNA is going to be harder. How much harder? Well, at a first pass, at a guess, you could say at least 1,000 times harder. How much longer will it take? Well, I don't know. How about 1,000 times longer? 1,000 times longer than 30 minutes is 500 hours. Oh, that's a lot of time. I want my cut to heal a lot faster than it could heal if I have to wait 500 hours for every cell division. This is a problem. Not only do we have the hurdles of these internal membrane-bound compartments to clear, but our genome is prodigiously large. And that brings with it its own set of problems. One of the ways we solve that particular problem of having a large genome and speeding up the amount of time it takes to replicate it is we divide our genome up into multiple chromosomes. So unlike prokaryotes that typically have just one chromosome, there are some that have two circular chromosomes, but that's sort of a footnote kind of thing. Unlike prokaryotes with effectively, let's just say between friends, one chromosome, we've got more. As humans, we have 23 pairs of chromosomes. We have 46 chromosomes inside of each of our nuclei. Part of that is so that it speeds up the process of replicating our chromosomes. You don't have to replicate all 3.2 billion off of one copy. You do it in chunks of 23, or 46 if you prefer, at a time. So there's that. But now this strategy of breaking up our genome into these individual smaller chromosomes brings another problem. And that other problem is we have to make sure that when our cells are dividing, that each of the daughter cells gets a perfect copy of each of those chromosomes. When one of your cells divides, again, you got 23 chromosomes. They're cleverly named. The biggest one is chromosome 1. The next biggest is chromosome 2. The fifth biggest, 5. All right? Doesn't do you any good if you make a mistake on one of those chromosomes. Say your cells are dividing and you did a really good job of getting a copy of chromosome 1 on each side of it. Each daughter cell gets one. You get a copy of chromosome 2. Hooray, good for you. You get 2 copies of chromosome 3 on one daughter cell and no copy of chromosome 3 on the other daughter cell. You know what? You're just plain old screwed. There's a clinical term for both of those cells. They're called dead, or at the very least, dead cells walking, right? The one without the information from chromosome 3 is going to be seriously compromised. There's stuff that it needs to do it can't do. It doesn't have the genetic instructions for. The one that got 2 copies of chromosome 3, unlike the prokaryotes, it's got a problem. It's got a gene balance problem. It's making twice as many copies of the proteins off of chromosome 3 relative to the ones from 1 and 2 and 4 and 5 all the way up to 22, right? It's got a problem. It's got a balance effect. Both of those cells have serious issues that effectively take them out of the gene pool. They're gone. They're not going to amount to anything at the end of this. So eukaryotes were complicated, or cell division is complicated. 2 things, more than anything else, speak to that complexity. We've got internal membrane-bound compartments. They get in the way. The nucleus, the nuclear membrane, for instance, in particular, obviously gets in the way of this kind of cell division. And our genomes are really long and cumbersome. They're large. And it takes a while to replicate them. And they're broken up into these smaller chromosomes. And each of those chromosomes has to be faithfully, perfectly transmitted from one round of cell division to the next. So all this while, lurking on the screen behind me has been a picture of a eukaryotic chromosome. This is mostly just to get us some nomenclature here. This is a chromosome, a single length of DNA that contains some portion of our genome, some portion of our DNA sequence. When cells are dividing, the chromosomes get replicated and they get held together at a centromere. You can see a picture of that centromere here. So what we see on this side is just a copy of what you see on this side. These are both chromosomes. When they're hooked together like this, they're referred to as sister chromatids. And when the cell divides, one cell's going to get one copy of one sister chromatid, its own chromosome, and the other cell's going to get the other sister chromatid, its own chromosome. Okay? And so they're just showing you here. They're helping you count noses, keeping track of the DNA molecules that are involved. So our chromosomes are going to be an issue. And here is just a cartoon that's giving you a sense of what's going on with the cell cycle. Again, we have to replicate those chromosomes. We have to do some DNA replication. When does that happen? Well, within eukaryotes, it's helpful sometimes to break it down into these different phases. The replication of a eukaryotic nucleus is what's happening during mitosis. That's this word here. So mitosis has a formal definition. It's a simple definition. Mitosis is the whole shebang associated with making a copy of a eukaryotic nucleus. Okay? That's all it is. Mitosis is the steps you need to go through, the process you need to go through to start with one eukaryotic nucleus and end up with two. That's what mitosis is, plain and simple. And when does mitosis happen? Well, there's a phase in the life of all of our cells that's called the mitotic phase, sometimes just the M phase. You know what's going on there? You're making copies of nuclei. You started with one, you end up with two. There are other phases in the life of a eukaryotic cell. Really, one other big phase. There's the mitotic phase, and then there's the time when you're not making copies of the nucleus, and that is called interphase. Okay? It's the time between the times that you're making copies of the nucleus. Obviously, there's a lot of attention that's being paid to this by the biologists who are studying it. This gets a lame name like interphase. This gets a fancy name like the mitotic phase. Interphase can be broken up into three parts. There's a G1 and a G2. G is short for growth. So this is the time when the cell's just hanging out, getting bigger, eating stuff, doing its metabolic processes, whatever its job for that particular cell is. That's what's happening during G1. It's growth. At some point, some cells make the transition from G1 to the S phase, and it's S for synthesis, specifically DNA synthesis. And it's during the S phase of interphase that cells actually roll up their sleeves and make a copy of their genetic instructions. Those 3.2 billion nucleotides of ours, that's when they get replicated. After the DNA has been replicated, cells will pass through something called G2, the second growth phase. What happens during the second growth phase? Well, pretty much what you might imagine. They may do some more metabolism. They may eat some more stuff. They may get a little bit bigger. Funny thing about interphase is that each of these steps, G1, S, and G2, are kind of variable in length from one cell and from one set of circumstances to another. Most cells park and hang out in G1. They never bother to replicate their DNA unless they get a signal that says, hey, buddy, it's time for you to divide. We need another cell just like you. When that happens, they leave G1 and they enter into the S phase. A cell, a brain cell, for instance, can be hanging out in G1 for decades. If you live long enough, maybe even a century, right? So G1 can last a really long time. The S phase takes some time. Again, we've got a lot of DNA that needs to be copied. If you put the pedal to the metal and you do it as fast as you can replicate your genome in a couple hours, right? So the S phase can be just a few hours, but it could also be drawn out over the course of days or weeks. This phase could be 2 minutes long. It could be 2 decades long. This phase can be a few hours long, could be a few weeks long. In G2, some cells park here so that they're ready to go through mitosis as soon as they get the signal. They need the division to take place really fast. They don't want to take the time waiting for the DNA to be synthesized. And so they'll park here, and some cells will park in G2 for years, waiting for the go-ahead to finish the job. Other cells rush through G2 and move in a matter of minutes from S to mitosis. But again, the focus for today is going to be on what's going on over here. It's worth mentioning, though, that there's other stuff, there's other parts to a cell's life. It's just we're focused today on how it is you do this hairy job of replicating a nucleus. So, mitosis. Mitosis gets broken up into a number of different subphases. I'll tell you, I think the single most important thing for me to communicate with you in this lecture is what I'm about to say. Don't get hung up on the details. It's perhaps the single most important thing for me to tell you. These names, prophase, metaphase, anaphase, telophase, right? These names are names that people, like you and I, people put on to this process. Living things don't give them any special significance, right? They all blend sort of one into another. What we're seeing here is a process. It's been convenient for cytologists, the people who study cells, it's been convenient for cytologists to refer and to talk about different steps in the process with these different names. But you're in for a world of unhappiness if you try to figure out the precise difference between anaphase and telophase, right? It's just part of a process. Cells don't have any light that flashes or a bell that rings that says, oh, we've transitioned from anaphase to telophase. It's just they all blend casually together. It's a continuum. It's a process. And so I think what's important for you guys to do here is realize that people do use these names, that we're talking about different parts of the process, but more than anything else, to appreciate what the process is. And then for shorthand, we can talk about what it means to be in prophase, and that just means to you, oh, that's part of the beginning of the process, right? We're early on in the process, as opposed to telophase, which is close to the end of the process, okay? So let's talk about what's going on. This is mitosis. Bottom line, we've got to start with one nucleus, and we're going to end up with two nuclei. Okay? Once we have the two nuclei, we can have a cleavage furrow that splits the cell into two cells. We'll get one nucleus into each cell, daughter cell, and we'll be happy. We will have succeeded in replicating, reproducing a cell. But it's a process. And what's got to happen in the process, I'll give it to you quickly here, and we'll go through it in a little bit more detail in the next couple sets of slides. What do you need to do to accomplish this business of replicating a nucleus? Well, there are a couple of jobs that have to be accomplished. I like to start with this one. This seems obvious to me. That nuclear envelope, it's got to go, right? So among the things that need to be accomplished is you better get rid of the nuclear envelope. The nuclear envelope starts out pretty tangible, right? Serious double membrane structure. Big deal. You can see it with a microscope inside of cells. So it starts out intact. How do you get rid of it? Little bit at a time, right? And the way you describe this to sound very scientific is you say the integrity of the nuclear envelope begins to be compromised, right? We start to break down the nuclear envelope. It starts to fall apart, is pretty much what's going on here. So at the start of the process, you should probably start to disassemble, break down the nuclear envelope. How about that? Not a bad objective. Seems like something that needs to be accomplished. That should happen at the beginning of the process. Now with that said, when it's all done, you'd better reassemble this nuclear envelope. So when we're all done, it's going to have to be reassembled. But at the start, we're going to break it down. Another thing that has to happen pretty early on is I've suggested that our cells have 23 pairs of chromosomes, 46 chromosomes inside of each of our nuclei. They're a long, tangled mess of spaghetti strings, okay? The way that they're usually deployed inside of our cells. Tangled up is an important idea here. But they have to be disentangled. How do you disentangle these long string-like spaghetti molecules? Well, the way that our cells pull that off is they condense each of those spaghetti molecules down. The chromosomes need to be condensed. Instead of being long and stretched out, they get pulled together into more manageable chunks that it's going to be harder to get tangled up one relative to another. So what do we got to do at the beginning of the process? We got to break down that nuclear envelope. What else do we have to do? We have to start condensing our chromosomes so it'll be easier to tease them apart. Okay? What else do you need to do? Well, ultimately, we're going to need to get our daughter chromosome, our daughter nuclei in their chromosomes, and we have to get the chromosomes to opposite sides of the cell so that we can do a cleavage furrow and get half of the genetic copies to one side and the other half to another. So we got to lay the groundwork. We have to set the stage to be able to pull those copies of the chromosomes apart, and that's where these guys come into play, these centrioles in the mitotic spindle. The centrioles start out right next to each other, but as mitosis progresses, they move to opposite sides of the cell. All right. So I've been talking with you about what happens at the beginning. Once again, at the beginning, the nuclear envelope's got to start to fall apart. And you can see they're showing you some little gaps in the membrane here. That's supposed to make you think the envelope is starting to break down. The chromosomes are starting to condense, okay? And the centrioles are beginning to move to opposite sides of the cell. That's a mouthful. That was what, 3 sentences, easy, that I've just said. Shorthand for all of that, prophase, right? That's what's going on at the beginning of mitosis. That set of things are all happening simultaneously. When those things begin to happen, we're talking about prophase. Your textbook takes this perhaps to extreme and suggests to you that you could break prophase up into even more phases if you liked. You could talk about prophase and you could talk about prometaphase. And all they're trying to do is tell you that as you progress through prophase, what happens? The nuclear envelope becomes less and less distinct. The centrioles are closer and closer to the opposite sides of the cell, and the chromosomes are even more condensed than they were at the beginning of prophase. Okay? You want to talk about prometaphase? You go right ahead. I'll be able to do the translation. Oh, you're talking about what happens later, further along in prophase. Not the beginning of prophase anymore, but the end of prophase. Other people will talk about early prophase and late prophase. I don't really care, right? It's a continuum, it's a process. I think the process is what's important. Prophase is just a word that we use to not have to say what's going on in the process at that point in time. It's a shorthand. But guess what happens? As you're progressing through mitosis, once you've done the business of getting rid of the nuclear envelope, once the mitotic spindle, and better, the centrioles are on opposite sides of the cell, and once the chromosomes are as condensed as they can be, those chromosomes line up, interestingly, on a line, a plate running through the cell. At that instant in the process, when the centrioles are as far apart as they can get, when the centrioles are as far apart as they can get, when the nuclear envelope is completely gone, when the chromosomes are as condensed as they can be, and when they're all lined up on one plate in the middle of the cell, that instant, as you're passing through that point in time, that instant we call metaphase. The rest of these phases of mitosis are phases, they're processes, they take some time. You're moving in a certain, that's an instant. Metaphase is bam, there was like a, have you gotten to metaphase yet, or have you not? There's no sort of like I'm a little bit pregnant kind of thing here. There's no like I'm almost at metaphase. If you're almost at metaphase, we don't call that almost at metaphase, we call that prophase. If you want to be really fancy, we call it late prophase or prometaphase, right? Have you passed through my metaphase, right? Have you gotten past that point in time? There's no such thing as late metaphase. there's the beginning of anaphase, right? So metaphase is unusual in this series because it's an instant. It's the moment when the chromosomes are as condensed as they can be, when the centrioles are on opposite sides of the cell, when the nuclear envelope is gone, and you got the chromosomes on this metaphase plate. Okay? What do you need to do in the process after that instant? Well, guess what? We got to start getting those chromosomes to opposite sides of the cell. The mitotic spindle needs to shorten. So we're pulling the chromosomes apart by shortening the protein rods that are connecting the chromosomes to the centrioles. That's the mitotic spindle. That gets shortened, and the chromosomes get pulled to opposite sides. If you want to be fancy, you say poles of the cell. So we got to separate out these chromosomes, one copy of each chromosome to each side. You know what else? No reason for the chromosomes to be condensed anymore. They can start to decondense. And may as well get a leg up on it. Eventually, you're going to need to make a new nuclear envelope around each of these clumps of chromosomes. You can start making a nuclear envelope again. That's what's going to happen after you've passed through metaphase. We got a word for that. Instead of saying all those things, we say you do anaphase. And when you've done that for a while, and you're getting pretty close to being done, the chromosomes aren't very condensed anymore. The nuclear envelope is starting to be pretty noticeable. The mitotic spindle is as short as it's going to get and even starts to go away entirely. You're almost done. That's telophase. That's the end of mitosis. And at the end of telophase, you know what you got? You got two nuclei where once you had one nucleus. Ta-da! That's what mitosis was. Again, we can break it up into steps and give each of those steps names, but the strategy, the process is pretty much straightforward. You could, if you like, keep your eye on the nuclear envelope. It needs to break down. It needs to be reformed. You could keep your eye on the centrioles. They need to move to opposite poles of the cell. And then their mitotic spindle needs to shorten to pull the chromosomes apart. It's another thing you could keep track of. You could keep track of the density of the chromosomes. They start out not that condensed. They get super condensed, and then they start to decondense such that when you're back to having two nuclei again, they are not that condensed at all. So maybe three things to keep track of. They're all working in sync, right? And they work out pretty much the way you would expect that they would have to work out for this process to work. I don't know that I need to say too much more than that. Don't get too hung up on what the difference between anaphase and telophase is. Right? I will have dozens of students in this class come to me within the next week and say, so what's the difference between anaphase and telophase? Right? I know it. It's coming. It happens every year, every time I talk about this. The difference between anaphase and telophase is just further along in the process than anaphase is. Right? That's all that's going on here. But is there like a new protein that's there? Are the chromosomes like 60% condensed and then they get to 70% condensed or 61%? Nobody knows. There is no precise definition. Cells make no distinction. There's no difference to a cell. There's nothing special that's happening in anaphase relative to telophase other than by telophase, you're getting closer to the end of the process than you were when you were back at anaphase. And when you were back at anaphase, you weren't that far away from what was going on at the metaphase instant in time. So this stuff actually happens. It happens every time our cells divide. You got a genome that's about 3.2 billion cells. You also have billions of cells. Okay? Those cells didn't come from nowhere, right? They came from other cells that were dividing. Your cells are dividing right now as we speak. Millions of cells are dividing every minute within you. This is going on all the time within all of your cells. And the cartoons that we had been looking at the previous slide, you get to see them here again, but they're paired up with actual photographs of cells going through the process. And let's see, it's actually going in this direction, right? So let's see what's going on. Where's the DNA? They've got a blue dye that's letting you see where the DNA is. They've got these spindles that are showing up here in red. Where's the DNA during G2? It's all bundled together. What happens in prophase? Well, the nuclear envelope starts to go away. I can't quite see a nuclear envelope here, but I can see the chromosomes are all clustered together. Do you think you can see little lines of chromosomes here, whereas over here that would be harder to make out? You know why? If you can see that from where you are, it's because the chromosomes are condensing. I can see it. Let's see if you guys can. So the chromosomes condense. The nuclear envelope goes away. And I think those are the centrioles. Yeah, there they are. Look at that. Those are two centrioles. They're starting to move apart. One there, one there. Eventually they're going to move here, and they'll separate. What happens a little bit further on in the process? Look at that. Now you can definitely see those chromosomes as individual squiggly lines. You know why? Because they're more condensed than they were here or here. There is no more nuclear envelope to speak of, right? Those things are all kind of poking out any which way. Here they're kind of still bundled together. The mitotic spindle, the centrioles, have moved to opposite sides of the cell. How about that? At metaphase, all the chromosomes line up. The mitotic spindle's on the opposite side of the cell. The chromosomes get no more condensed than that. And then in anaphase, the mitotic spindle starts to shorten, and it pulls the chromosomes to opposite sides of the cell. The nuclear envelope starts to form around both of those clusters of chromosomes again, and by the time you're at telophase, the mitotic spindle is as short as it's going to get. The chromosomes are no longer distinct. You can't see individual lines very clearly anymore. There's a nuclear envelope that's encapsulating each of them. And one extra thing, this business of cytokinesis is in play. Notice how this cell is getting kind of pinched in the middle. That's because there's a cleavage furrow there. It's constricting. It's a ring. In the bag of goo, there's a ring of fibers, and that ring of fibers is contracting, and it's pinching that one big bag of goo into two smaller bags of goo. And we'll see a picture of that in a little while. All right. How many times am I going to talk you through this? You've got to be wondering. I'm wondering at this point. You know what the next 6 slides are? It's like each of these steps over and over again, all right? I'm sick of saying what's going on in these steps. I've told you, right? I don't know how much more I can say. If you need to have it repeated, look at the Tegrity recording, rewind about 10 minutes, and you'll hear the whole thing one more time. If you want to see zoomed in pictures of what's going on here, that's what these next slides are showing you. But one more time, right? You start at G2 of interphase. That's the end of interphase. Check it out. The centrosomes are clustered right next to each other. The DNA is all squiggly. I haven't said anything about the nucleolus today. Remember, the nucleolus is the place inside of the nucleus that ribosomes get made. You don't need to be making ribosomes when you're replicating chromosomes, when you're replicating nuclei. One of the things that also happens at the beginning of the process is the nucleolus becomes, it breaks apart. You don't see it anymore, and it doesn't reappear again until the end of telophase. So the nucleolus goes away. That's something new that I haven't said before, but the nuclear envelope looks pretty good here. No big problems. You got a plasma membrane out here. The chromosomes are duplicated, but they're all stringy and squished together. You start mitosis. Prophase begins, the centrosomes start to move apart, and you begin to lay these spindles out, these protein rods that are going to hook up to the chromosomes in metaphase. The chromosomes get more condensed, the nuclear envelope starts to fall apart. By the time you're at the end of metaphase, the mitotic spindle is pretty much fully made. The centrosomes are on opposite sides of the cell, and the chromosomes are pretty condensed. At metaphase, those chromosomes line up on the metaphase plate. At anaphase, they get pulled apart. The things that holds the two sister chromatids together breaks. That centromere breaks, and the centrioles start to pull them apart with their mitotic spindle. And when you get to the end of mitosis, the chromosomes begin to get all diffuse again. The nucleolus starts to reappear. A nuclear envelope begins to form. The mitotic spindle's gone. And you do this business of a cleavage furrow to give you two cells, each with a nucleus, where once you had just one cell with a nucleus. Whew. That's it. That's what's going on with mitosis. What's the word you would use to describe this? Isn't that cleavage for you? I mean, that's what a cleavage furrow looks like, people. You got a cleavage furrow right there. That's where the name comes from. That's why people talk about this as a cleavage furrow. I've already told you what's happening is there's a ring of fibers, microfilaments, in the middle of the cell that just gets smaller and shorter all the while. As it does, it pinches off that one big bag of goo into two smaller bags of goo. And we can take pictures of the process and see that's just what's going on. At a molecular level, a cartoon is showing it to you. There's a cleavage furrow. All right, so that was cytokinesis, right? Cyto, we're talking about the cytoplasm. Kinesis, movement, we're moving the cytoplasm to two different cells. That's cytokinesis. Oh my gosh, more pictures of the same thing. Now this looks a lot like what you were seeing in lab, right? These look to me like onion cells going through that have their chromosomes stained. They're going through the different phases of mitosis. I don't think I need to tell you about this anymore. You guys could tell me because you've seen these with microscopes and you've heard me prattle on about this for the last half hour. You start with prophase, you do that part of the process, you get to metaphase. Look at that, they're all lined up on the metaphase plate. They start to get pulled apart after metaphase, that's anaphase. The difference between anaphase and telophase is in telophase, you're starting to see some cytokinesis taking place. A cell wall is getting formed here. The chromosomes are fully pulled apart, they're decondensing, you're getting close to the end. Seriously? We got to look at each of these now? Who set up this PowerPoint presentation? This guy needs a serious talking to. He's wasting your time. All right? But oh my gosh, there's a picture of prophase. All right? What's happening in prophase? Oh, it's the beginning of the process. What has to happen at the beginning of the process? Get rid of the nuclear envelope. Lose the nucleolus. Start getting your spindle set up. Move the centromeres to the opposite sides of the cell and start to condense the chromosomes. Fine. End of prophase. Okay, that's happened for a while. When it's happened for a while, you get to metaphase. The chromosomes are as condensed as they can be. The spindle's intact. The nuclear envelope is completely gone. And you're ready to start pulling these chromosomes apart at the beginning of the end, which is anaphase. And at the end of the end, which is telophase, the chromosomes are completely separated, and you're separating the cells into two parts. What do you think the odds are? Is there another set of pictures that I'm going to go through with you here? Is there that was 4, right? Is there going to be a 5th? I sure hope not. Maybe. I don't know. This is a repeat, so it's not as bad as going through the whole process again. But let's zoom back out. We've been down in the weeds for the last 15, 20 minutes talking about this stuff. Let's zoom back out and remember this is part of a process. Okay? The process that we've been really focused on here is mitosis. Mitosis is this business of replicating a nucleus. We got a problem. Our problem is that we only have one nucleus. We want 2 nuclei. How do we solve that problem? Mitosis. Okay? So that was a process, but it's just part of a bigger process that's going on within the life of a cell. For some cells, they're going mitosis pretty much nonstop. There are cells that are giving rise to red blood cells. We're cranking through red blood cells all the time, right? We have to have these cells dividing pretty much nonstop. The cells, the stem cells that are giving rise to our red blood cells are in mitosis pretty much every minute of every day that they're not in S, right? So they're busy synthesizing DNA. They spend very little time in G2. They spend very little time in G1. It's all about making baby nuclei and copying DNA. right? So there's some cells that are full tilt. All their time is spent here and here. There are other cells, like brain cells, that park here. That's interphase. And when you're talking about interphase, that brings up an opportunity to talk about some interesting opportunities. Let me put it to you this way. You know, cancer, it's bad, right? Nobody wants to have cancer. Nobody aspires. Oh, I'm going to get cancer someday. Can't wait. Looking forward to that. No, that doesn't happen. At the heart of it all, you know what cancer is? The formal definition of cancer, it catches all of the many different flavors and varieties of cancer. At the heart of it all, cancer is uncontrolled cell growth. When you got some cells that are just dividing all the time without regard to signals they're getting from their neighbors, without regard to what's best for the organism, they're just about dividing and making copies of themselves, that's a cancer cell. That's going to give you a tumor, right? Not good. Why is it our cells aren't always just making copies of themselves? Well, there's a short answer, and the short answer is there are checkpoints that each cell has to pass through to be able to get the go-ahead to go to the next step in the cell cycle. Those checkpoints have been pretty well characterized. The people who characterized them actually got a Nobel Prize for figuring this out. And what cancer cells are doing is they just blow right past these checkpoints. Uncontrolled cell growth. Normal cells have 3 checkpoints they have to work through. And that's kind of a nice system, a nice design to the system, right? Because if a cell gets a mutation that causes it to be able to move past one of the checkpoints that it would have normally stopped at, it's not that horrible because there's still 2 other checkpoints that are going to keep it from dividing any further. Not bad, huh? Each of these 3 checkpoints is independent. You got to have multiple problems to convert a normal cell to a cancer cell that can clear all of these checkpoints. Where are those checkpoints? Well, one of the big checkpoints is called the G1 checkpoint. And what it's doing is it's keeping cells in that G1 phase. What has to happen is really simply this. For a cell to get to the point where it starts to replicate its chromosomes, it needs an affirmative signal. It needs something, some molecule needs to come, at least one needs to come and tell it, hey buddy, your time has come, get ready, you're going to divide eventually, right? Start making copies of your DNA molecules. It needs that message. It just doesn't, of its own accord say, I've been in G1 long enough, I'm bored, I'm going to move on to the S phase. That's not how it works. You need an affirmative signal, something that taps that cell, communicates something to it, and tells it, okay, the time has come for you to progress from this part, this kind of metabolism in life, to the stuff that's on the other side. By the same token, even though a cell has replicated its DNA, and even if it's hung out in G2 for a long time, that cell needs another affirmative indication that tells it, all right, you've replicated your DNA, your time has come, do the nuclear division trick. What's that trick called? Oh, it's mitosis. Start mitosis. There needs to be another affirmative poke, something that tells it, okay, start doing nuclear division. Start prophase in cytology terms, right? So there's another checkpoint. And somewhere between anaphase and telophase, there's still another checkpoint that needs to be cleared before mitosis gets completed. So there are 3 checkpoints, major choke points on the path here for this cell cycle. For a cell to become a cancer cell, it needs to be able to clear all 3 of these in a way that is no longer responsive to the controlling mechanisms that normal cells are responding to. All 3 of those have to end up being a problem. So what's happening? What are these signals that the cells are getting? Each checkpoint, each of those 3 checkpoints has its own signal, right? Let's talk about the one that gets you, well, there's 2. There's a protein called mitosis promoting factor, and there's another thing called cyclin, a protein that's present inside the cell. As time goes on, cyclin concentrations increase, more of that protein gets made. And if that happens in conjunction with an increase in the presence of the protein called mitosis promoting factor, you end up getting a pass through that second mitotic checkpoint. So both of those things have to have happened to be able to make it through the end of mitosis into G1. And you can actually, within a cell that's actively dividing, you can keep track of the concentration of those 2 things, and that's what they're doing here on this graph, and they're showing you that as those 2 things reach their critical thresholds, the cell gets the message, okay, it's okay for me to move on. And how is that controlled then? Well, cyclin's getting accumulated during interphase. It binds to this guy, those 2 things, the mitosis promoting factor, the cyclin, and the thing that it binds to together are the mitosis promoting factor. As you go through mitosis, cyclin gets degraded, and you have to start making it all over again. The CDK obviously is not being changed. It's there all the time, but cyclin levels need to increase to give the cell that signal that it's time to go through the end of mitosis, or actually here it's getting through the end of G2. All right, and I tell you what, that was fairly painless, right? Even with the 5-6 times of seeing all the pictures of prophase and anaphase and metaphase. Here's the deal. We'll come back here again on Thursday, and I'll talk with you then about not just mitosis. There'll be a quick refresher on that, but then we'll talk about meiosis. And you know what's going on with meiosis? The trick is this. You start out with a nucleus, and you need to end up with gametes. You need to end up with egg cells and sperm cells. One extra round of nuclear division without DNA replication. That's it in a nutshell, but that's the star of the show for Thursday. See you guys then.